Hello and welcome to today's Connect webinar brought to you by the International Society for Stem Cell Research. My name is Jack Mosier and I'm the Scientific Affairs Manager at the ISSCR. Today's webinar is the first in a three-part series that will focus on the tissue-specific stem and progenitor cells known as MSCs. The series will highlight the history and biology of the cells, their experimental use, and their clinical use. Today's presentation in this series is entitled Tissue-Specific Stem and Progenitor Cells known as MSCs, the history, origins, and functions. Before I introduce our speaker, I would like to direct your attention to the Ask a Question box at the bottom of your screen. At the conclusion of the presentation, we will take questions from viewers. However, feel free to enter questions into this box at any time during the presentation. We will answer as many of them as time permits. Our speaker today is Dr. Pamela Roby. Dr. Roby holds many titles, including Chief of the Craniofacial and Skeletal Diseases Branch, Co-Coordinator of the NIH Bone Marrow Stromal Cell Transplantation Center, Chief of Skeletal Biology Section, and Acting Scientific Director of the NIH Stem Cell Unit. In addition to these responsibilities, Dr. Roby also runs a very active laboratory at the National Institutes of Health. Without further delay, I would like to turn the presentation over to Dr. Roby. Dr. Roby, thank you for being here today.
Thank you, Jack, for inviting me to tell the members of the ISSCR about tissue-specific stem progenitor cells, cells that are very commonly known as MSCs. So the points that I'm going to cover today is to talk about where the concept of a tissue-specific stem progenitor cell came from, using skeletal stem cells and bone marrow stromal cells as an example, and I'm going to talk about their developmental origins. I also want to highlight the role that SSCs and BMSCs play in disease, and to suggest that perhaps other tissue stem progenitor cells can be used in the same fashion, and then to talk about their use in tissue engineering and regenerative medicine. So what exactly is a tissue-specific stem progenitor cell, which so many people will call a mesenchymal stem cell? And I think in order to understand where this field is now and how it got there, we have to take a look back and perhaps take a look forward. And so the original notion of a tissue-specific stem progenitor cell comes from a number of very seminal pioneers in our community. First, Alexander Friedenstein, working in Russia in the late 1960s, who later teamed up with Maureen Owen, working in the United Kingdom in the 1980s, and then the contributions of our dear friend and collaborator, who unfortunately passed away about a year ago, Paolo Bianco. And so what the original concept was from Alexander Friedenstein and Maureen Owen is that in bone marrow there is a non-hematopoietic, very rapidly adherent clonogenic cell that attaches what Alexander called a colony-forming unit fibroblast. And when they attach, they begin to proliferate and form these beautiful colonies. He also called these cells mechanocytes. This is based on the fact that they produce extracellular matrix, and they migrate, and they are able to contract matrix. And so what he found is if he took these cells and he put them into a diffusion chamber and transplanted that into the peritoneum of a mouse, what he found was that these cells would form cartilage in the interior of the diffusion chamber, most likely due to the anaerobic environment, and that they formed bone in the part of the chamber that was closest to the external environment. So this is a closed system transplant. If he put these cells into a gel foam or a collagen sponge and transplanted them underneath the kidney capsule, what he found is they were able to make a complete bone marrow organ with bone, stroma, and adipocytes of donor origin and hematopoiesis of recipient origin. And so this is a very defining feature of the bone marrow stromal cell and the skeletal stem cell. They are the only postnatal cell type that are able to support hematopoiesis. And this was the original concept. However, this concept became altered. It was thought that, well, if bone marrow stromal cells can make mesodermal derivatives such as bone, marrow fat, and cartilage, they could also make other mesoderm derivatives such as muscle, tendon, ligament, and other connective tissues. And so this actually got altered even further by saying, okay, if they can make mesodermal derivatives, perhaps they can make all connective tissue derivatives, and they were subsequently named mesenchymal stem cells. However, this is problematic because if you look at the classic definition of what mesenchyme is, as defined by developmental biologists, mesenchyme is an embryonic connective tissue, and that during embryonic development it gives rise to not only connective tissues, but also to blood and blood vessels. And currently there is no rigorous proof to date to show that any postnatal stem cell has the ability to form all three of these tissues and to form them functionally. And so the term mesenchyme is really meant only to be used in respect to embryonic development. In addition, these two alterations, alterations 1 and 2, are not developmentally sound. And so in order to take a look at this and understand where these cells arise from during development, we can take a look at what happens after gastrulation. After gastrulation and formation of the 3 embryonic germ layers, the mesoderm layer becomes further specified. And so what we see is that there are at least 3 different types of mesoderm. There's the paraxial mesoderm, intermediate, and the lateral plate. And the paraxial mesoderm will go on to segment, and the most dorsal part of that segment in the somite will form dermis. And then beneath the dermal forming layers, there is the muscle forming layer and the tendon forming layer. And also you get muscle cardiomyocytes formed by splanchnic lateral plate mesoderm. Now, if you go into the development of adipose tissue, surprisingly, the developmental origins of adipose is not as well worked out as for dermis, muscle, and other mesodermal derivatives. However, it is now known that neural crest can give rise to facial adipose tissue, and it's generally regarded that paraxial mesoderm is the source of adipose in other parts of the body, but this is not as well worked out. In terms of bone formation, what we know is that the facial bones are formed by neural crest, that the ventral part of the somite, called the sclerotome, gives rise to the axial skeleton, and the somatic lateral plate mesoderm gives rise to the appendicular skeleton. So for bone formation, at the very least, there is no common cell that is formed during embryonic development that gives rise to all bones in the body, and this is a very important point. So if we continue along with the development of the bone marrow organ, what we know is that mesenchyme undergoes condensation, and that in the interior of that embryonic condensation we get the formation of cartilage, and this is the patterning of bone formation. As the bone development continues, what we see is that the interior cartilage begins to undergo hypertrophy. This hypertrophic process calls in blood vessels and brings in cells that will degrade the hypertrophic cartilage and bring in with it the osteoprogenitor cells that will form the periosteal collar and the primary spongiosa. And then as development continues, we see formation of bone in the epiphyses, again through a vascular invasion process. And we also see the appearance of marrow adipocytes. This is a very, very late occurrence. It occurs postnatally and not during embryonic development. So what we see here is that there is a timed emergence of the skeletal lineage that we have during embryogenesis. We have embryonic mesenchyme, which condenses to form cartilage. Most of the cartilage that is formed during embryonic development undergoes hypertrophy and is replaced by bone. Some embryonic cartilage is maintained as articular cartilage. As bone development continues, we have formation of bone marrow stroma, which supports hematopoiesis, and then postnatally, when hematopoiesis and bone are formed to the level that's necessary, that stromal cell will take on fat and become a marrow adipocyte. So, as we know from the work of Alexander and Maureen, that postnatally, bone marrow stromal cells reform skeletal tissues because they contain a subset of multipotent skeletal stem cells. And so if we take a single colony and we divide it into different parts and we put that single colony into anaerobic conditions such as micromass cultures, they form cartilage. If we take the rest of that colony and we put it onto an appropriate scaffold and put it in vivo into immunocompromised mice, and we use those because we're using primarily human cells, we get formation of a ectopic ossicle, a complete bone marrow organ. The big point here is that this is by clonal analysis. This has been demonstrated on individual colonies and not by the population as a whole. And what most people will not tell you is that only 1 out of 5 of the original CFUFs, the colony establishing cell, are multipotent and are skeletal stem cells. And it's also important to note that these cells support not only bone formation, but they control bone resorption, and this makes them very, very central for skeletal homeostasis. So the take-home message here is that all skeletal stem cells are colony-forming unit fibroblasts, but not all colony-forming unit fibroblasts are actually stem cells. So because of their very unique ability to form bone and to support hematopoiesis, a great deal of effort went into trying to identify the cell surface markers for bone marrow stromal cells. Unfortunately, these markers are not specific. They will identify virtually any adherent fibroblastic population of cells, and really cannot be used to identify a population as a population of stem cells. Another big issue about the use of these markers is that their value really is only in terms of freshly isolated cells. That if you apply these markers to cells that have been cultured, we know that many of these markers change with time and culture. So the real value of the markers should be on isolating freshly isolated cells. However, there was another alteration to the concept of a bone marrow stromal cell and a mesenchymal stem cell proposed by the International Society for Cell Therapy. And they published a position paper for the criteria for defining multipotent mesenchymal stromal cell, which is, again, not an appropriate term, mesenchyme being embryonic, stromal meaning that not all cells that are stem cells in connective tissue are stromal in nature. And what their recommendations were that in order to classify as a MSC, the cells had to be plastic adherent in standard culture conditions, that they must express CD105, CD73, and CD90, and that they must differentiate into osteoblasts, adipocytes, and chondroblasts in vitro. And this is a major issue because many of the in vitro assays are either artifactual in nature or are misinterpreted. And you'll hear more about that from Mustafa Kassem when he presents on October 26th. So, based on these criteria, a huge literature blossomed identifying mesenchymal stem cells in virtually every connective tissue based on plastic adherence, based on expression of 3 nonspecific markers, and based on the fact that they underwent some artifactual differentiation in vitro. And this has really confused the field tremendously. In spite of the fact that none of these markers used individually is informative in terms of a tissue-specific stem progenitor cell, one marker did emerge in the use of human cells that was very informative, and this was a report published by Sacchetti and Professor Bianco in Cell 2007. And so I should also mention CD146 is a very good marker for human cells. For murine, there are other markers that are emerging that are very useful, in particular leptin receptor. And if you take a look at papers from Sean Morrison's group, there are a number of very interesting studies identifying leptin receptor as a good marker for murine cells. So in terms of CD146, what exactly is it? Well, it's a member of the Ig superfamily. It's also known as MCAM and MUC18. And it also binds to the alpha-4 laminin, which is expressed by endothelial cells and is thought to be a marker for cell association between different cells. Now, if you take a look at what CD146 means in terms of human bone marrow, we found that if we sort human bone marrow and removed the CD146 positive cells and isolated the CD146 positive cells, that those were the clonogenic cells present in bone marrow. CD146 negative cells were non-clonogenic. If you look at where it is expressed in bone marrow, you can see that it is found on the subluminal side of sinusoids, and this is what we would typically call a pericyte. In addition, Stan Gronthos and Sung Tao Shi also found that CD46 in human dental pulp was found associated with blood vessels. So if you were to take bone marrow stromal cells and you also isolate dental pulp cells by the same cell surface phenotype, and you do the in vivo transplantation assay, what you find is that the human bone marrow stromal cells make bone and they support formation of a hematopoietic marrow. If you take human dental pulp MSCs and do the transplants, you see formation of beautiful globular dentin, which is what is exactly found in a secondary tooth, and they reform a pulp-like complex. So here you have an example of two cell types that have the identical cell surface phenotype, and even when you think about bone versus dentin, they make almost the same bone matrix proteins. But when you do the appropriate, rigorous, gold standard in vivo transplantation assay, you find that they make completely different tissues. So we wanted to go on and continue along that vein, and we did a study where we isolated cells from a variety of connective tissues using the same cell surface markers of CD45 negative, CD34 negative, CD146 positive. And we isolated those cells and grew them and characterized them under identical conditions. And this is a study that we recently published in Stem Cell Reports. And so what you see is if you take cells from bone marrow, muscle, periosteum, cord blood, we also looked at amniotic fluid cells and adipose tissue. They have the exact same cell surface markers. So you would not be able to distinguish these cells based on cell surface markers. But if you do transcriptome analysis and then PCA analysis, you see that these cells separate based on their tissue of origin. So while their cell surface is very similar, this is due to the fact that they are fibroblastic in nature. And if you scratch below the surface, what you see is that they reveal the tissue that they have come from. So for example, the muscle cells have very low levels of the myogenic transcription factors. Bone marrow stromal cells have the transcription factors associated with bone formation. So this is a very clear demonstration that while the cells are very similar, they are very different in their transcriptome. And we went on to analyze these cells by using rigorous differentiation assays. So in the first case, we proceeded with our standard in vivo transplantation assay, where we attach the cells to a scaffold and put them into immunocompromised mice. And what you see when you use bone marrow stromal cells is you get complete reformation of the bone marrow organ. You can see the bone is very nicely illuminated by polarized light, and you can see that there is hematopoietic support. If you do the same exact assay with muscle cells with the same cell surface phenotype, grown under the same conditions, you do not see any formation of bone at all in these transplants, and you only see formation of very sparse connective tissue. On the other hand, in terms of looking at muscle differentiation, the in vitro assay is the gold standard for demonstrating inherent myogenesis. And that is because if you use the in vivo transplant in conjunction with an injury, myoblasts will fuse with virtually every cell type. They are not picky about who they fuse with. And so the in vitro assay is the gold standard without myoblasts, additional myoblasts, to see that the cells can fuse on their own. So we do this by taking the isolated cells, expanding them in 20% fetal bovine serum, and then we switch the culture conditions that are supportive of myogenesis. And in doing this assay, you can see that the cells from bone marrow, periosteum, and cord blood do not spontaneously form myotubes, but the cells from muscle very nicely form myotubes in vitro. In terms of their ability to self-renew, which is a defining feature of a stem cell, we previously provided evidence for self-renewal of bone marrow skeletal stem cells in Sacchetti Cell 2007. In the current paper, we provided evidence of self-renewal for muscle cells. What we did was we took cells plated at clonal density, combined them and injected them into a muscle, an injured muscle. We then enzymatically released the cells from the muscle and isolated human cells using human-specific antibodies. We expanded them into culture, and we again checked to see if they were expressing CD146 and CD90, and they were. The CD146 positive cells were plated, and we were able to identify colonies. The CD146 negative cells did not form any colonies. We then took those colonies and looked at the expression of their markers again, and we found that they maintained expression of CD146, and in addition they also had expression of CD56, which is a marker for very early myoblastic cells. So this is evidence of self-renewal, and this is the type of evidence that is lacking from many different populations of cells that are called mesenchymal stem cells. Previously, we also reported that bone marrow stromal cells are pericytes, and that they would be able to form associations with endothelial cells. And so we asked the question of whether or not These cells, isolated from other tissues by the cell surface markers 45 negative, 34 negative, and 146 positive, could also form pericytes. And we did this by using a very specific assay. What we did was we took GFP positive cells with the desired phenotype. We mixed them with Huvex, and then we transplanted them underneath the skin of immunocompromised mice along with Matrigel, growth factor reduced Matrigel. And what you can see after 3 weeks with bone marrow cells is that you have the formation of microvessels, where you have the endothelial cells on the inside, as you can see by the endothelial cells on the inside, as you can see by the CD34 positive staining, and the cells forming pericytes labeled in green. And you can actually see red blood cells flowing through these vessels, indicating that these microvessels are functional. And we were able to demonstrate that for bone marrow cells and also for muscle cells. And you can see on the bottom part of the slide that very beautiful microvessels with red blood cells inside of them are formed in this in vivo angiopoiesis assay. So this indicates that not only are bone marrow stromal cells able to form pericytes, but also cells from other tissues with similar cell surface markers. So this kind of leads us to try to explain why connective tissue cells have such similar cell surface characteristics, but such different differentiation properties. And what we think is happening is that during embryonic development, blood vessels do not have pericytes, and that makes them extremely labile. And so as they are growing into a developing tissue, which has already committed to a specific lineage, they hijack whatever cell they run into and incorporate that cell on its cell surface as a pericyte. And these pericytes remain quiescent along with the endothelial cells and the blood vessels until they are liberated by either the need for tissue turnover or due to injury, at which point they reveal their true differentiation capacity. So we can see this happening during bone development. We also can see this happening during the formation of muscle, and we also see this happening during formation of dental pulp. So the common theme here appears to be that when the blood vessel is growing in, it recruits a cell to become a pericyte. That cell is held quiescent, but then undergoes differentiation after injury or during homeostasis. So many people say, okay, so why do we question the term mesenchymal stem cell? What is in a name? And my answer to that is that I think that we need to be rigorous, that during embryonic development, there's no common mesenchymal stem cell. So why would we expect there to be a common mesenchymal stem cell in the postnatal organism? Secondly, we now know that clonogenic cells from many tissues are CD146 positive pericytes, but they are not equivalent in their differentiation capacity. More tissues should be tested, but again, they should not be given a common name if they don't have the same differentiation capacity. It's also very important to note that pericytes are not a lineage, just as mesenchymal stem cells are not a lineage. You cannot pinpoint a single cell during embryonic development that gives rise to all pericytes in the body. And for this reason, I think it's very important for us to think about changing the terminology, and that the cells should be named based on their tissue of origin. And so you may ask, why does this matter? Well, it matters in terms of basic biology, because currently everyone is trying to fit their cells into the mesenchymal stem cell box. But it's kind of like trying to fit a round peg into a square hole. there's no reason for us to try and force muscle-derived cells to form bone, because it doesn't happen in vivo unless you're hit with a huge amount of bone morphogenetic protein. And if we were to focus more on the inherent differentiation capacity of these various different cell types, then we would be able to better utilize them in tissue engineering, and we would understand more about their biology and how diseases affect the biological processes that they undergo. So it matters for basic biology. It also matters for stem cell therapies, and we will talk about that a little bit later. So the next thing I want to talk about is the role of these tissue-specific stem and progenitor cells in disease, and I'm going to use skeletal stem cells and bone marrow stromal cells as an example. So our original hypothesis was that because SSCs and BMSCs are very central mediators of bone homeostasis based on the fact that they control bone formation and resorption, we hypothesized that any genetic mutation, which would be an intrinsic change, or any change in the microenvironment that alters their biological activity, will result in a skeletal disease or disorder. And for example, I'm going to show you fibrous dysplasia of bone. This is caused by activating missense mutations in GS-alpha, which leads to overproduction of cyclic AMP, and you get formation of a very abnormal bone and marrow fibrosis. So the concept here is that the skeletal stem cell is the unit of postnatal skeletal disease. And the proof of this principle came by, again, using our in vivo transplantation assay, where we took the bone marrow stromal cells from normal marrow, transplanted it into the mouse, and we got the formation of a normal human ossicle with bone and marrow being supported. When we used the mutated cells, what we found is that it completely recapitulated a bone that was completely reminiscent of the native lesion in situ. That is, that it formed woven bone, and it did not support the formation of hematopoietic tissue, but formed a fibrotic tissue instead. So here we have an example of a piece of human bone in a small animal. And so these mutant ossicles can be used to try and design new therapies for the treatment of this and potentially other skeletal diseases. So for example, we can work on ways to correct the mutation by using CRISPR-Cas9, or to silence the mutations, or to develop treatments that use small molecules to inhibit the activity of the mutant GS-alpha. And this, in fact, is being done by my colleague Michael Collins, working with NCATS, is working on developing small molecules that will inhibit the mutant GS-alpha. And so one of his tests will be to grow the human diseased bone in the mouse and then look for efficacy of the small molecules. So it's possible that other tissue-specific mutated stem and progenitor cells can be used in a similar fashion with the appropriate assays. For example, if we wanted to look at cartilage diseases, we could use the pellet culture. If we wanted to look at muscle diseases, we would use the in vitro myotube formation assay. And that these assays could be used to try to have a better understanding of the pathogenetic mechanisms that leads to various different types of connective tissue diseases. In the last part, I want to talk about the use of tissue-specific stem and progenitor cells in tissue engineering and regenerative medicine. And at this point in the game, I'd like to point out, and we seem to have a problem with my slide. I'm sorry, what you should be seeing on the left-hand side is the human body. And what the slide is showing is that right now there are only a very few stem cell therapies in which the stem cells themselves have been shown to replace a connective tissue. And examples of this are corneal where limbal cells have been used to replace the cornea. We know that bone marrow transplantation is very efficacious in terms of restoring hematopoiesis. We know from a number of small clinical studies that bone marrow stromal cells can be used very nicely to regenerate new bone, and we also know that epidermal cells can be used to replace skin. However, this is really not what most people are using mesenchymal stem cells for. And if you go through the literature, and this is an example of bone regeneration. In my institute, we are now working on a clinical protocol to use autologous bone marrow stromal cells to regenerate jawbone defects that are caused by the removal of benign tumors of the jaw. And we're hoping to be able to get this study underway in the very near future. However, if you look in the literature, many of the studies that are using all kinds of mesenchymal stem cells are using them for the so-called paracrine or immunomodulatory or immunosuppressive effect. And they are being used to treat a really long list of diseases, different types of fibrosis, different types of ischemia. If you look at clinicaltrials.gov, what you will see is that there are over 600 clinical trials in which MSCs of all kinds are being used. However, there are a lot of questions about this, and again, I apologize. On the right-hand side, you should again see a picture of the human body. If these cells are systemically infused, they are very rapidly entrapped in the lungs, and they are very rapidly destroyed and disappear. Even upon local injection, the cells do not survive for extended periods of time. And so for this reason, there's a big question as to the use of cells in this type of therapy. So the questions that come up from these types of studies are that so far the paracrine and immunomodulatory and immunosuppressive effects of these various different types of cells is not stem cell dependent. So, for example, no one to date has isolated a clonal population that is proven to be a stem cell that is in exerting these paracrine effects. So far, all of these effects are based on the population as a whole. There may be subsets that are not stem cells that mediate these beneficial effects, but this has not really been documented well to date. The other big issue is that the rationale for the treatment of many different kinds of injuries and diseases and disorders is often lacking, and this is a major issue that needs to be addressed in terms of looking at the efficacy of these clinical trials. Many of the trials have not really measured appropriate outcomes, and they're not well reported. So if you go to clinicaltrials.gov and you look to see what has actually been published, you would probably be very surprised that very little has been published. There is one study that was published by Nobar, who did a meta-analysis on all of the trials that used MSCs for treatment of cardiovascular disease. And the conclusion of that paper was that the more significant the outcome measures, the less there was an effect. And so this is the type of meta-analysis that is going to be needed in order to try and demonstrate whether or not there really is efficacy. And then the other major issue here is that in most cases there has not been rigorous determination of the mechanisms of action in most cases. It's very hard to understand how the cells that get trapped in the lungs or how cells that rapidly disappear upon direct injection can be mediating an effect. It makes the mechanism of action somewhat obscure. So what this leads us to really is that we need to sell help, not hope. This is an article that was written by Paolo Bianco and Doug Sipp in 2004. And the basic gist here is that if we focus on the true nature of the cells that we are working on, we may be better able to treat diseases. And instead of trying to hype our cells as being miracle workers when we really don't know that much about them and about their biological activity. And I'm sure that you will hear more about this when Megan Munsey and Mark Young give their presentation on November 30th. So I think it's really important for us to look forward and to start to call the cells by their appropriate names, which would include their tissue of origin, and that we really need to understand better the basic biology of these cells and not try and make all of these cells fit into one box, because it is not helping us figure out more, gain more knowledge about these cells. So in summary, what I have told you, and I hope I've convinced you, that bone marrow contains a subset of bona fide skeletal stem cells. Skeletal stem cells are pericytes, but not all pericytes are stem cells. Other tissues have stem progenitor cells that are pericytes, but you have to keep in mind that pericytes are not a lineage. They are an accident of development. They are a cell that happened to be in the path of an invading blood vessel. Skeletal stem cells and bone marrow stromal cells play a big role in skeletal diseases and can be used to model human disease, and it's likely that tissue-specific stem and progenitor cells could be utilized in the same fashion. We can use these cells to regenerate functional bone, but other usages are not well characterized to date, and we really need to work on that as well. So I would like to acknowledge again Alexander and of course Paolo for all of their contributions to the field, to the members of my group, Sergey, Natasha, and Mike Collins has his own group, but has worked with me on fibrous dysplasia. My former fellows that have contributed to the studies that I have presented here, and to Mara Riminucci and Alan Boyd as longtime collaborators. And at this point I will stop and ask if you have any questions. You can also contact me. My email address is pretty simple, but if anything occurs to you or if you'd like more information, please don't hesitate to ask.
Thank you, Dr. Early, for that illuminating presentation. We have a few questions that have come in, and we'll take more as we go along. Feel free to submit those. So the first question is, what is the frequency of CD45 negative, 146 positive cells in the different tissues? Do you have, did you assess that?
Yes, I have, but I can't recall the numbers. If you plate a clonal density for a connective tissue, so for example, bone marrow stromal cells, the frequency of CFUFs is between 10 and 50 per 100,000 cells. And out of those 10 to 50 CFUFs, 5% are multipotent. Now, the problem with the other connective tissues is that they are not heavily contaminated with blood. So clonal density for cells that are enzymatically released from connective tissues is 1.63 to 16.3 cells per centimeter square. And when we plate those cells at that density, we get roughly, and don't quote me on this because I would have to look it up, I think it's roughly 50 cells per 100,000 mononucleated cells.
Thank you for that answer. This kind of goes in hand with that, and it's what is the what percentage of CD45 146 positive cells give rise to multilineage colonies. You mentioned previously that the frequency of bone marrow multipotent progenitors was 1 out of 5. Wondering if you had data to that effect for the 146 positive cells.
Right. It's the same. It's about 1 out of 5. There's a little bit of variation. I mean, doing clonal analysis is very time consuming, and we could only handle about 25 clones at a time. It would be nice to do many more, but so you see a little bit of variation from one donor to another, but it's approximately 1 out of 5.
Thank you for that answer. Going back to the CD45 negative 146 cells from the muscle, what extent, if any, do they overlap with the satellite cells, the residual stem cells in the postnatal muscle?
Yes, they are in an anatomically different location. So the satellite cell is underneath the basement membrane in contact with the myofiber, whereas the CD146 positive cells are pericytes in small blood vessels. So the question is, are they related? And it's a possibility that, again, it's just a matter of location, that they are the same cell, but one happened to be hijacked by the blood vessel and the other one grew along with the developing myofibril.
Interesting point. Thank you. I think I have one last question here, and this is a little bit off topic, but I'd be curious to hear your thoughts on it. You mentioned that, the question is, many of the therapies offered based on, quote, MSCs are from the fat, specifically the stromal vascular fat. And I was wondering if you or anyone that you know of has kind of looked in that tissue and has assessed the, quote, MSCs and what developmental insights that you have on them.
Right. So we have done a few studies on fat-derived cells, and when you do the in vivo transplantation assay, they don't make bone, they only make fibrous tissue. If you do the chondrogenic pellet culture, they make a fat ball if the fat cells are really, really good fat cells. But what most cell populations that have been given to us to test, what they do is they make a ball of dead cells. In terms of adipogenesis, if you put them in hydrogels, they seem to form fat. I should mention that the scaffold we use is hydroxyapatite, which is really geared towards bone formation. But in hydrogels, fat cells, or adipose-derived stromal cells, which is what I call them, will form very beautiful adipocytes. Now, in terms of their other uses, that is really a question. And unfortunately, there have been 2 patients in Florida that were treated for blindness by the injection of adipose-derived stromal cells into the vitreous humor, who instantly went blind. So I think that we have to be very cautious. Now, I'm not sure I completely answered the question. I think I've kind of forgot the second part of it.
Well, I think you answered it. It was just how would you characterize the cells, the quote MSCs that are purportedly used therapeutically in the stromal vascular fat? And it sounds like largely you answered that.
Okay. So again, if there are any others that have questions out there, just drop me a line, and I can send papers or answer you to the best of my ability. I appreciate the opportunity to have the chance to give this webinar.
Thank you, Dr. Roby. This concludes today's presentation. And again, I would like to thank Dr. Roby for taking the time to give us this perspective on skeletal stem cells, what are commonly called MSCs, or mesenchymal stromal cells, although she mentioned the issues with the use of that nomenclature. I would like to remind viewers that today's presentation, like all of those on ISSCR Connect, will be archived, so please feel free to view it again at your convenience. And on a programming note, as Dr. Roby mentioned, there are 2 other upcoming presentations in this series. On October 26th, Dr. Mustafa Kassem will give a presentation entitled Know Thyself, Rigorous Assays for the Characterization of Tissue-Specific Stem Progenitor Cells, known as MSCs. And on November 30th, Dr. Megan Muncie and Mark Young will give a presentation entitled Understanding the Clinical Uses of So-Called MSCs. Thank you for attending today's webinar, and we hope to see you at these future webinars. Thank you, and have a good day.